So in this next video, what we are doing is that we're trying to carry out laboratory studies on reproductive biology of fish. And when we talk about reproductive biology of fish, we are talking about certain things such as the sex determination. We want to determine the sex of the fish. We also want to see what is the stage of the development of the gonads. Is it immature? Is it developing? Is the gonad ripening? Is it ripe? Is it ripe running? Is it spent? Is it recovery and resting? You understand? So these are things that we have taught in class, which we can also check in some other videos. So what we are going to do is that, still working by the specimen. The specimen, we already did the length and weight measurement. So what we want to do now is to check for the sex. There's some fish species that you can easily identify the sex without even opening up the fish. Examples are the catfishes, like Clarias grappinus, and also these chrysisis. Clarias grappinus is what? It's a freshwater species. But since we are just working with these ones, which are marine or brackish water species, if we have chrysisis negogitatus, that is silver catfish. It's a brackish water species. We can't be able to tell, because one thing you notice about the male is that there's a way on the underside. There's the genital papilla that we will notice to be able to determine that this is a male and this is a female. But for species like this, it's not totally easy as such to identify. So what we will do, at least at this basic level, is that we will take the fish. This is a Carans chrysos. We will take our dissecting kit, like I've said the other time, and I removed maybe the scissors. Then I will look for the anal point, and I will insert the scissors. Okay. Oh, sorry. This is the anal point. Okay. So you will now try to cut. Like when women cut clothes. You lift it, you cut. You want to cut it open. The same way, when you want to determine the food and feeding in fish, especially when you want to check for the stomach content. So because of the nature of this fish, some other fishes don't have this kind of thing that will be disturbing. So what we'll do is that once we have done this, we'll open up the fish. Okay. Because of the nature of this fish, I need to have a bigger scissors. So when the fish is opened up, I want to examine the items in the fish. Now, this particular one, the issue is that for the reproductive study, once you have opened up the fish very well, you will see the gonads are actually two, whether male or female. Now, looking at this, and the gonads, because of most times fishes have what's called cloaca. The cloaca is the external aperture through which about 3 different ducts coming from the fish, from different organ system in the fish, they release to the external. What are the ducts? You have the excretory, you have the reproductory, and you also have the one from the digestive tract, the alimentary, the intestine. Let me put it that way. The intestine to release the what do you call it, urea or whatever. It's the ducts with they all fuse at the anal region, from internal to the anal region, and they release through the cloaca. Now, For in this case now, what we are looking for is the one for reproduction. The one for reproduction. So in some fishes is very, very clearly displayed, in which you will see two like this particular one now. I'm trying to locate the second gonad. The second gonad. I think the flesh. So the gonads, well, maybe because of the stage of development, like this particular object, this particular fish, I would say it is not showing that it's not showing that it's a female. Okay, I'm coming. Let me see the way the gonads are. You know, when you work with, depending on when you work with different specimens. So what you do, what you see is that if it's a female, it will have two sacs, one on this side, another one on the other side. And these sacs, you will take note of the stage, the color, to know which stage of development it is. Now, for a fish like this, don't be surprised. It could just be at the stage of immaturity, or at the stage of spent, or at the stage of recovery. You know, we said it in class. At the stage of recovery, it will appear as if it's immature. But really, it's not immature. What happened is that the gonad has passed through one cycle of what? Reproduction, and is recovering and preparing for another. So the gonads will be two, one on this side and the other one on the other side. And if it's a female, it will be sac-like. The gonad, the ovary will be sac-like. If it's a male, it will be tube-like. Then you now know, take note of the stage. Now, if it's a female, you know, this is just a specimen. By the time we look for bigger specimen, peradventure we might meet a female showing us clearly the features of the ovary. Maybe when we meet that along the way, features of the ovary, and we will not check that what stage is this ovary. So when we get the ovary, for instance, now, if it's a male, of course you will record it. For instance, like this fish now, you can record, except you have read in literature for this fish, to know what is its size at maturity. So if you don't have that information, you may assume that a fish like this, you may say it's immature. But another thing is that, well, maybe due to the exposure, you know, depending on the timing the fishermen caught it and the rest, some spoilage will have occurred to the internal organs. And so these are some of the reasons why you may not be able to at times find clear, maybe because they are already disintegrating. So it's good to work with fresh specimens so that you can easily do together. So for instance now, for this fish now, for somebody who is just starting, you may just record, for instance now, you can say the sex is immature. Immature. You can say immature. Immature means, because I don't want to assume for now that it is resting. Do you understand? It is when you go back and check literature. That's why it's always good to start with few, so that you will be able to correct whatever along the way. So for something like that, you go back to literature. What is maximum size? So this either immature or at resting stage. That's what I can place this fish. Whether it's male or female. For immature, you cannot tell it's male or female with your naked eye. You can only say immature. It's acceptable to write immature. Since what are you checking it? You are checking it based with your what? Naked eyes. So you can put immature, but when you go back and check literature, you might be able to know that this, because it's looking more like immature or resting stage to me. All right. So, but if it has egg, what we do is that we cut the gonads. We cut the two gonads at the point of connection with the body. You cut them and you bring them to the measuring, to the what? To the petri dish. You want to determine the weight. So we have ovary weight there. Do you see ovary weight? So you bring it here. You know, you zero it, because you have put this thing will contribute to the weight. So you zero it, you tie it, so it becomes 0. So when you put the ovary there, you will record the weight. Measure the ovary, whether it is mature or not mature. But the one, by the time we want to now do fecundity, our emphasis will be on the matured ovary. Do you understand? So with the mature ovary, for instance, if you are working with the mature ovary, after measuring the ovary weight, and you record the gonad stage, even for everyone you record gonad stage, you record the ovary weight. You know, you may wonder, sir, well, what about male? For male, we don't bother ourselves recording any weight for the for the testes. We don't bother recording. So you record that. Now, but for fecundity, your emphasis is on, is it a ripe gonad? Ripe female gonad. So we will take that. What we'll do is that we will now, I will teach you maybe later, maybe when we encounter a gonad with egg, how you are going to be able to check and estimate the fecundity. Fecundity is what? Is number of ripe eggs in a mature female fish before what? Before the next spawning season. So that's what will be demonstrated by the time we have a ripe egg or ripe gonad. Is that noted?
So,
thank you.